That's where we should go, as opposed to just go blindly and say we should carve out this as a national park. Here is a very systematic, a very scientific way of designing national park, a conservation area. And I think this kind of tool gives you a powerful information to do that. The other thing that came off, which was not our original objective, but we were so interested in this project, was, like I mentioned, Chitwan National Park, 420 samples screened, 240 of them were tigers. We wanted to know what were the rest of the other samples. So we did DNA barcoding on those samples, and we found out there were 72 of them were of common leopard. There were leopard cat. There were species that we hadn't cataloged before, maybe, perhaps. Yeah? So by virtue of the fact that you are looking for tigers, because tigers are charismatic, they're not the sole animal that we should save, but you know, they are charismatic and they bring resources. Very, very powerful. And if someone is interested in tiger biology and see where they move, who they interact, who they breed, their offspring, we can all do that with this kind of study, with this kind of technology. And I mentioned we built the wildlife database. And this is a tool that is so powerful that everybody wants to have a piece of it. And I've presented in many different areas and countries, and everyone looks at it. Everything is searchable, right? So I'm just going to show you, demonstrate to you. I want to look at Chitwan National Park, and you can use any of these parameters. And then it gives information about general information, field information. And if you look at it, it automatically generates a map, and that's where it was located. Bunch of tiger skins, or skins. You bring it to our lab, we do DNA extraction, we find out whether it's tiger or not. If it is a tiger, if it is in our database, we can tell exactly where it's coming from. No one has it. No other countries has it. So we're very, very proud of what we did. And all this software was developed here in Nepal. And so we're very, very proud of that. The other thing that we did was we wanted to work very closely with the Department of National Park. We wanted to make information readily available to them whenever they needed it, as opposed to calling us and asking for it. So what we ended up doing was setting up this presentation. A lot of this information and more detailed information is in that report that you have received. If you have any questions, let us know. In the acknowledgement section, I cannot thank the previous USAID mission head, Dr. Kevin Rushing, enough, because the first conversation that I had with him, and I mentioned about our idea, he readily accepted it. Had it not for him, we would not have been talking about this today. And I'm really, really appreciative of his thinking and responsiveness in doing this kind of work. Our main collaborator, our main collaborator, she couldn't be here. Our co-PI couldn't be here, but they have sent a small video, and I would like to play that so that we are all familiar with them, and they have contributed greatly to make this a global system.
But it's been a great pleasure to work with the Center for Molecular Dynamics Nepal and all the collaborators on the USAID-funded Nepal Tiger Genome Project. During the last 2 years of this project, I've had the pleasure of hosting Nepalese scientists in my laboratory for training and visiting Nepal twice for additional training, and also getting to see the amazing countryside and habitat of the Bengal tiger. So together, we have learned much and accomplished many things. One of the greatest challenges to preserving tigers in Nepal and across their range is the lack of scientific data to inform conservation and management decisions. Our collaborative project was designed to help address this gap by developing non-invasive genetic sampling tools and increasing local capacity to obtain information on the population size and genetic structure of the Bengal tiger. Our effort has been very successful and will benefit the conservation and management of Nepal's unique biodiversity in multiple ways for many years to come.
With the successful implementation, I want to say that we not only fulfill our desired objective with the project, but also tried our level best to integrate a new technology like DNA-based to complement our past conservation efforts with endangered species like tigers.
Chitwan National Park. And also,
queries or questions. We have Halsey and D&D here to answer those.
I don't read this one. Hi, I am Naresh Trivedi. I work with NTNC and I'm working with tigers since last 20 years in the rare landscape. I have some questions. And okay, first of all, I would like to thank CM Kim for the good job and excellent presentations. I have one question on that. You collected 1,200 scat samples and you did analysis for 700. So what was the basis of selecting 700 samples out of 1,200? That is one that I want to know just for them. And I was actually wondering about the result of Chitwan National Park, where you found 72 different individual tigers. Out of them, 52 were males.
yeah.
Actually, my actually head is striking. Are there 52 male tigers in Chitwan National Park? Because, okay, we can compare this because from the camera trap photographs also, we can identify most of the animals, actually sex of the individuals, because you can see the sex organs on the camera trap photographs. In 2009, I was involved in Chitwan and I was leading the Chitwan team, landscape, this camera trapping operations with Kanchan and your team actually. So I have a big question, whether there are 52 male tigers in Chitwan, if that for the tiger population is true. That is one. And then in habitat suitability map that you presented, I don't see that was properly designed. Actually, the modeling has some problem. Because for the tigers, so you need to check on that modeling actually. What parameters you were taking for modeling? So that is I think you need to revise that one. And then, okay, this is a good initiation. So that this is basically based on the 2009-10 fieldwork data. The tigers. 2011. Okay. So in Chitwan National Park alone, what we found from the camera tracking data is there is a 60% turnover of the tiger individuals, tigers. So this is nice start, but we need to continue to track on what you were saying. You can track the mother of the animals and then the relations of these individuals. So if we carry this genetic work very intensively, then you can track it. Otherwise, this is only the one-time work. And then you cannot really say anything about the tigers because there is 60% tiger turnover in the Chitwan National Park. So I mean, this is a good work, but you need to tie up it with the field sites, I think. So thank you very much for this, and yeah, we can discuss.
So of course, we were limited by our budget. Initially, we thought we would only do 700, but we ended up getting more than 700, and so we had to kind of actually had to prioritize our resources where that needs to go. And for the selection of those 770, we tried to take as much of a representative sampling from each of the national parks or areas that we collected.
The question is, how can you say the representative? Because I
From GIS.
Saura,
Yeah.
tiger from the Saura, actually, visit the tiger.
Yeah, I understand.
60 kilometers.
Right.
Because from our satellite planning of the tigers, what we know is that the tiger in Saura, for example, moves overnight to the Madi, and then it moves to the tiger. So it actually travels around 80 kilometers in 2-3 days. So if you say that the tiger scar sample from the Saura is different from that of the tiger cubs, that may not be in the field.
Right.
So, and then my question is, if you say you could have done the analysis for 1,200 samples, and then you could have come with the total numbers. Anyway, this is a good initiative. We should appreciate this. Because as a scientific community, if we raise the question, then we need to actually justify it with the strong statistics.
I agree with you. I think we should do 1,200, all of it. We couldn't. And then also I have to mention that this is a pilot research project to build a baseline information. Yeah. So, and then the second question I think you had was about the habitat suitability map. That's just a concept. That's not an actual really map. But what we are saying is we know the tiger points, and then we can do this, and then we can extrapolate. So the map that you're looking is just a conceptual map, just not the real map. And the third question that you had was?
And the population, actually, I was amazed with these 52 males.
Oh yeah, that's right. So actually, that's a good question because several people have raised that question. How come you're seeing more males than the females? And it turns out they have done a similar study on one of the cats in North America, and they're seeing the same thing. And there are various hypotheses, and again, it's just the hypothesis, right? Usually, the likelihood of finding male tigers is high because they roam around, they have bigger territory, number one. Number two, males also, because they are territorial, don't cover up their scats, so the likelihood of finding those scats is higher. So it's probably all statistical bias because of their behavior. But no one has done any kind of extensive research to find out whether it is actually a statistical bias or is it just the way it is. To figure this out, you probably have to do several rounds of survey, and you get more tighter, more high resolute information about the tiger numbers. Again, I would like to mention, this is a baseline information. This is how we start. Proof of concept can be done in Nepal. Well, should we do it next year? And day after? Right? So that's...
Just a small clarification. You said that 15 by 50 degree size we use for camera traps, not true. We use for open survey. Fair enough.
Okay.
Anyway, but we use a 2 by 2 size for camera trap. And I'm really concerned about the heterozygosity matter.
Mm.
The results show that it's 0.5. Yes. So it's really critical. Actually, we should need to think differently. How we can mix up the population? How can we increase the population? The quality.
Yeah.
So that's really. And the other thing regarding the habitat suitability. And in 2008-9, we did a camera trap survey in Chitwan, but at that time we excluded the Churia part, right? Then we didn't have any information about on the Churia side. And 2010, we again repeated the camera trap, and at that time we included the Churia. Then we found a very good population of tiger in Churia also. So I think we need to consider that matter also. Just saying the suitability map is, we need to refine it.
Sure. Again, that's just the concept. That's just
really good,
nice
job.
Genetic analysis for the time conservation. I just two questions. So based on this the scat sample the analysis, and by using the different analysis, so if were you able to localize the territory, because the territory, because this tiger is very and well known for its territories. So were you able to locate each tiger's the territory is number one. And then the number two is based on the cytochrome B analysis, were you able to analyze the tiger with the specific the variations and then the distribution based on the cytochrome B analysis, which is normally used for the interspecific analysis. So
well, to answer visual, again, goes back to the question of have we done enough rounds of it? Have we covered it all? Are we statistically confident that 90% of the tiger scat population is being covered by analyzing more samples? But to answer your question, yes, we can do that. Second part of your question regarding species identification. For tiger, we took a portion of cytochrome B region, which is very, very specific to tigers. So it will only amplify the tiger samples. And this is the work we followed from CCMB, actually, a lot of these things paper. So we have some correlated sequencing validation for it, and it's a good result for the species identification. Usually on the mitochondrial DNA, on the cytochrome B part, there are less variable within the species and variable between the species. And that's why we target mitochondrial DNA segments for species identification. And for the other species that we have identified, we targeted a certain portion of cytochrome B, and then PCR'd it, and then sequenced it, and did the blast on NCBI to figure out what species they are.
Studying anything on disease or something like that?
Right, so that's actually a good question. That's, as far as the prey species, we only looked at or ungulates, right? We just looked at the carnivore species. And also the assay that we used could also identify other species as well. Disease is the next thing we need to study on these scats, whether we will be able to identify viruses or bacteria on these scats. It's important for a couple of different reasons. Important because disease, for example, canine distemper disease, which is well known to kill tigers in captive population in Germany and other areas, is caused by a paramyxovirus. And we don't really know in the wild tiger population whether it's prevalent or it's found. But we do very well know that these canine distemper viruses are found in domesticated animals like dogs that are around the vicinity of the tiger habitat. So there is this likelihood of this virus going back and forth between wild animals and domesticated animals and human beings to that extent, right? So it's important that we study that. The other important part that we want to study diseases is we want to do a parallel study.